Welcome to this video. So today we're talking about biogeochemical cycles and how we can use nifH genes and other genes like that in order to understand more about what these genes are doing in the environment. And in order to do this, you usually have to do some level of cloning or metagenomic analysis. So this video is just about cloning and how that would look and how you'd interpret the data. So the first thing we're going to do in this exercise is cloning and using the nifH marker gene to study um, nitrogen fixation. So what we're trying to do is uh, looking at the biogeochemical cycles, the marker genes, and trying to understand how it is would look at each specific cycle by just investigate one marker gene. So the target gene we're using is nifH, and this is a marker gene that is used because it encodes a conserved component of the nitrogenase and is widely used to monitor nitrogen fixation microorganisms. So that is bacteria that are basically doing this. And you can do a little study for yourself to see what this actual enzyme looks like. And the objective here is fivefold. So, what you should be able to do, and if you can't do this, go through the exercise again till you can do it. That's the point of this exercise. You should be able to explain the role of nifH genes in biological fixation. You should be able to describe how environmental DNA can be used to detect nitrogen fixing microbial communities. That is from different environments like a river or soil or something. You should be able to design a conceptual cloning workflow for a nifH gene fragment from soil, sediment, or water sample. You should be able to analyze sequence data to infer diversity from nitrogen fixing organism, and you should be able to evaluate how nifH data can improve understanding of the nitrogen cycle in an ecosystem. So if you notice here, these things that you're supposed to be able to do after doing this exercise, they go up in different levels of difficulty. You should be able to just explain. So if somebody asks you, well, what's the role of nifH genes? You should be able to say it, explain. You should also be able to describe. So that is just being able to show that you understand it. You should be able to design. So coming up with an actual solution. If you're trying to clone a particular gene, you should be able to show the workflow, how it is you go from the beginning to the end. And then you should be able to actually look at sequence data to actually get something from it. And finally, you should be able to evaluate, think about it. What does this mean in the bigger scheme of things? So the scenario you're given is a hypothetical environmental sample from a wetland, um, and agricultural soil, mine impacted soil, and hot spring mat, and also a cyanobacterial bloom. Let's change that. And this comes up at the very end. So that's the exercise you're doing right at the very end. But before we get there, you need to go through this whole process so that you can actually fulfill this task. And your task is to design and interpret a workflow showing how a nifH gene fragment could be cloned and used as a molecular marker for nitrogen fixation. So the first part is a conceptual background. You should just start here and start answering these questions. What is biological nitrogen fixation? I want you to look into that and think about the actual reactions that are involved. And why is this enzyme nitrogenase important? So, again, as I told you earlier, look into this enzyme and do a little background reading about it. And then number three, why is the nifH gene used as a marker? So when you look at the enzyme, look at the component which is nifH. And then how does nitrogen fixation influence soil fertility, aquatic productivity, and global biogeochemical cycling? So again, do a little bit of reading to see how this fits in. The key idea here is that the presence and diversity of nifH sequences tells you something in an environment. It indicates the potential for biological nitrogen fixation. So in the previous lecture, we talked about metaproteomics, if you remember, and how you compare that with metatranscriptomics and metagenomics. So each type of omics tells you a different thing about that environment. But here, what we're talking about is that the presence of these genes and you're detecting them from the DNA, it tells you about the potential for biological nitrogen fixation in that environment. So, and I think you understand this now, that the gene detection alone does not prove actual activity. You need the other levels of the omics to do that. You need metaproteomics, especially, that tells you what's really going on. And metatranscriptomics tells you whether that gene is actually actively being transcribed in that environment. So the metagenomics just tells you whether it's there or not. So expression data, nitrogenase assays, or isotope-based measurements would be needed to confirm actual active fixation. So we use the DNA at a certain level in the analysis. It's not everything. And then part B, we look at the cloning workflow. So you should be able to draw this workflow after you're done and understand why it goes like this. Why do we start with the environmental sample? Why do we then go to DNA extraction? Then we amplify the nifH gene, and then we ligate this into a cloning vector. Then we transform it into host cells. Then we screen the colonies. Then we get the plasmid sequence and then check in a database and then do our ecological interpretation. So in this little exercise, we're looking at this section where you're actually cloning. I just looked at this part where you're actually cloning particular genes that are nifH genes into your plasmid. So at the end of this stage, you should be able to talk about the output. So if you're to cover this column here, you should say, okay, if it's environmental sampling, the purpose of this is to capture the microbial diversity. But how does that look? What is the output? It's a water sample, or it's a sediment sample, it's a soil sample. Then the next stage of DNA extraction. What's the purpose of this? We're trying to recover the DNA in the community. And what is the actual output? What you get in your hand? It's environmental DNA. nifH amplification. What are we doing here? We are trying to target the nitrogen fixation marker in your test, in your PCR reaction. And what you're going to get in your hand is marker gene amplicons, and so on. So that is what you should be able to do. If you're given this table, you should be able to actually say what the outputs are. I could go on. Um, if you're looking at the cloning stage, why are you doing a cloning? You're trying to preserve individual gene variants that you find from the environment. And what you're going to get clones. So when we talk about clones in this exercise, this is what comes out of this. When you get this PCR product and you actually put it into a plasmid, that is a recombinant clone. Then in the screening phase, why are you doing this? In the screening phase, you're trying to identify positive clones. And what you're going to get out of this is a clone library. So, so it's going to be a library of all the clones that are positive. Then in the sequencing stage, why are you doing this? You're trying to determine the gene identity. What is actually the sequence that you cloned? So that is your nifH sequences. That's what you're going to get. And they're usually in your FASTA format, right? And then in the bioinformatics stage, why do you do that? You're trying to compare the sequences, and the expected output, you're going to get some kind of an alignment or a picture which shows taxonomic or functional relationships that gives you that insight. And then finally, the interpretation stage. This links genes to the nitrogen cycle, and this gives you a conclusion to the whole ecological study. You can understand more about the specific genes that are doing this job in an ecological setup. So in part C, we're looking at the bioinformatics interpretation, and I would like you to use NCBI to obtain nifH sequences of your own. So just go there, find a nifH sequence, and then you note its similarity and the environment where it came from. So that is similarity to some of the sequences from your colleagues. And then you submit your sequence using the Google form, which is here. And I think I have it here. I haven't shared this. Ooh, well, I don't know if it will show, but the Google form looks like this. So at the end of it, you'll be able to just upload your sequences or paste them and then answer a couple of questions. Whoops. Yeah. So now consider a clone with the nifH gene insert. Follow these steps and draw a vector map so that you show such a clone where you have nifH cloned into PUC18 using EcoR1 and Hint3 sites. So I've got here 8 steps for you to follow. If you follow these step by step, you should come out with a nice vector map that shows your nifH gene annotated nicely in there. So after that, you follow exactly the same steps to include your own nifH gene, the one that you got from NCBI, and you create a vector map of that, and you upload both of those to the Google form. And then you have to look at the some hypothetical sequences to understand how it is you actually look and understand what they're telling you. So, how to interpret the sequences from multiple clones? Consider the data from 5 clones called nif01 to nif05 that all have nifH genes from different organisms cloned into PUC18. And these are the clone IDs. And this is the closest match that you got from your blast analysis. And this is the environment where they're coming from, and this is the similarity that they have. So the question here is, when you look at something like this, how do you interpret it? So let's look at these questions. Number one, which clones might suggest nitrogen fixation going on in the bacteria? So out of these, you'd look and say, okay, which ones actually have the ability to do this? And you might have to look into literature. You might have to look into the actual genes that each organism has. Is, for instance, like this one, this particular clone, it's got 89% similarity, but look where it's from. It's a methanogenic archaeon, so it's an archaea, and it's from wetland sediment. So if you know those two details, you can do a bit of digging and see if it fits. Is this a possible nitrogen fixing bacterium? Well, it's, think about it. It's an archaeon. Let's move on. Which clones might represent uncultured or novel diazotrophs? So again, you do a little bit of literature review, but you can look at each one of these and see what kind of functionality is it known to have in the literature. And this can inform your decision on what you say about this. And is it really new? Is it found in the literature already? So when you do your literature search, that will come out and you'll be able to determine that. What does the presence of cyanobacteria nifH suggest about nitrogen input in aquatic systems? So again, you do a little literature research about it and then see how that fits in. But you can see that it's got high similarity. And why might low similarity sequences be important in metagenomics? What do such sequences tell us? So this is coming uh, uh, to understand why both types of sequences are important. Sometimes low similarity sequences are there, and you're going to have to understand what they mean. High similarity sequences are similar to what you're expecting, basically, and they sort of tell you a positive story. But with low similarity sequences, they could tell you something like maybe a different type of way of doing the same job. So you could have enzymes which are sort of doing the same job, but they're not, they represent something new. There's a whole bunch of reasons why that could be. So your job is to think about that one and offer me some explanations. Then finally, it's part D, where we look at the biogeochemical cycle interpretation. So you should be able to construct this simple diagram where you show this flow, and then you annotate where in this whole cycle does the nifH gene, the product, where does this fit in? So draw your cycle and then put something where it says this is where nifH actually sits in the cycle. Upload this image to your Google form, and then discuss with your peers how can cloning and sequencing nifH genes help us to understand whether an ecosystem depends on free living, symbiotic, cyanobacterial, or archaeo nitrogen fixers. So think about that. That's a question which will definitely come up at some point, and you should be able to answer that one after you've actually looked at all these. And then you're going to submit a lab report which has these components, and this is how it's going to be marked. And that sort of ties up your learning about the whole process of going into environment, picking up a sample, using PCR to amplify the biomarker gene that you're interested in. In this case, it was nifH. Even determining which gene to actually target. So when you look at the whole cycle, like if this wasn't nitrogen fixation, if it was like sulfur cycling, how which part would you target? And this question allows you to sort of do that thinking. So after you've done all that, it should be very easy to put this together, and that would explain how we'd actually do it in the lab. Okay, I hope that was clear. If there are any questions, you can reach out. Thanks.